I would like to thank the organizers of the GFN for inviting me to present this video, which summarizes research undertaken by myself and by my colleague Sebastien Soulet. I will focus on preclinical studies using the InExpose manufactured by CIREC. It is a commercially available computerized system specially designed to generate vape aerosols and deliver them to exposed cell lines and rodents. Practically all studies using the InExpose claim that their experimental results on cells and mice show high likelihood of serious biological harms from vaping. As an example, one study, the authors exposed pregnant mice and found adverse effects in their offsprings. The press release extrapolated these results to human pregnancy. It was quite scandalous. To probe these worrying outcomes, we looked systematically at preclinical studies using the InExpose under the same aerosol-generating experimental conditions. We did a literature search that found 40 articles on same experimental conditions. The articles, some provided sufficient information, some insufficient information, but we managed to synthesize it. The default device used by the InExpose is a third-generation tank, the EVIC Mini. It is a high-powered device, a sub-ohm, and the InExpose restricts the puffing of this device to an airflow between 1 and 2 liters per minute. This is appropriate for low-power devices running below 20 watts. We became suspicious when looking carefully at the promotional video of the InExpose. At the beginning of the experiment, the liquid in the tank was colorless, but at the end of the experiment it acquired a brownish color. This is a signal of pyrolysis of the cotton wick, and it clearly shows there is overheating. The authors reported very different parameters, all taken from the EVIC screen that you can see below in the right. Voltage, power, temperatures in these ranges. However, we have to calibrate that. We have to verify if these values in the screen of the device had something in common and if it matched laboratory tested values. This is the result of the calibration, and I summarize it here. All experiments were conducted by puffing the EVIC Mini with a 0.15 ohm coil at power levels between 40 and 45 watts with airflow rate of 1 liter per minute. We replicated the puffing to verify if it was done under normal or overheating conditions. We also quantified the presence of toxic byproducts, mostly aldehydes in the aerosol. And this is what we found. It was actually puffed, the device, under the overheating regime. If the authors had wanted to obtain an aerosol without overheating, they should have puffed the device between 14 and 31 watts. Regarding the toxic byproducts, at the level of 40 to 45 watts, we find a 20-fold increase in toxic byproducts. The authors claim their experimental outcomes predict harmful effects in vaping. However, all the studies exposed cells and rodents to aerosols that were generated under overheating conditions, loaded with excess toxic byproducts, and the puffing was done in a way that is unrepresentative of consumer usage. I have to say something about the device. When the EVIC Mini was released, it was a technologically advanced product, among the first devices allowing for user-controlled temperature or power mode. However, since 2020, it has become an obsolete relic that almost no vapor uses. It is very difficult to find one in retail shops. Therefore, it is questionable to use this device as a standard default for studies published so recently between 2020 and 2024. We identified 105 research projects funding the 40 studies. The total funding is well over $10 million US, and it mostly comes from US government sources. Are there more preclinical studies with same methodological problems as the one we found? Yes. We found over 67 studies not using the inexposed but with the same problems. This means that there are likely much more studies under this condition. Thanks for watching.